Meiosis is a cell division process first observed in 1876 by German biologist Oskar Hertwig, who saw the process taking place in sea urchin eggs. It consists of one cell which divides twice, each time dividing into two cells, to make a total of 4 haploid cells in the end. These 4 cells are gametes, essential for human reproduction, and each have 23 chromosomes. Essentially, the entire process aims to reduce the number of chromosomes. In fact, meiosis literally means a lessening in Greek. Why is reducing the number of chromosomes so important, you may ask? Well, if 2 humans with 46 chromosomes each reproduced without a reduction in chromosomes, the embryo of the baby would contain a whopping 92 chromosomes, double the necessary number, resulting in severe genetic conditions. Therefore, meiosis is extremely crucial in the continuity of the human race and its diversity. Before a cell actually starts meiosis, it spends most of its life in intraphase, just like before mitosis. To review, intraphase consists of gap 1 phase, S phase, and gap 2 phase. In gap 1, cells go supersize. They grow much larger and create proteins and organelles. RNA, centromeres, and other components of a cell are replicated, and the cell moves on to S phase. In S phase, the DNA is synthesized. The DNA molecules, in the form of double helixes, unwind like unzipping a zipper. The 2 single strands are then utilized almost as templates to form identical strands. In gap 2, the cell continues to grow even bigger and finishes producing any molecules it needs to prepare for division. Now, let's move on to the actual process of meiosis and then cytokinesis. Firstly, there is Prophase 1, in which the nucleus begins to disappear and the centrioles move to the poles of the cell, releasing spindle fibers. The nuclear membrane also disappears. Duplicated chromosomes pair and a process called crossing over takes place. Basically, 2 homologous chromosomes stack on top of each other and exchange segments, resulting in 2 chromosomes which each have a different combination of genes, leading to genetic variation. Next, there's metaphase 1. In metaphase 1, the spindle fibers attach to the chromosomes, which have now aligned next to each other in the middle of the cell. In anaphase 1, the chromosomes are pulled to opposite poles of the cell by the spindle fibers. 2 key things are important to note in this stage. The first is that the sister chromatids remain attached at the centromeres and are not separated as a result of being pulled. And the second is that rather than all of the maternal chromosomes going to one pole and all of the paternal chromosomes going to the other, they are divided randomly in a process called independent assortment. This will ensure that each haploid cell produced through meiosis will contain genes from both the mom and the dad to ensure genetic diversity. In telophase 1, the homologous pairs of chromosomes at the poles of the cell are encased by 2 new nuclear membranes. Shortly after, cytokinesis happens and the cell is split up into 2 new cells. As you know, however, the goal of meiosis is to produce 4 cells rather than 2, so the process is only halfway done. All of the following steps happen in both of the daughter cells. The first one is prophase 2, in which the centrioles duplicate and the nuclear membrane breaks down once again. Next, there is metaphase 2, in which spindle fibers form and the chromosomes line up single file this time in the middle. Essentially, metaphase 2 plays out the same as metaphase 1, except with one difference being the fact that the chromosomes line up single file rather than side by side. In anaphase 2, the chromosomes are split apart into 2 sister chromatids each, which are now considered chromosomes of their own. Notice that although the number of chromosomes doubles, the number of chromatids stays the exact same. Independent assortment also takes place again, so that each cell can have a combination of maternal and paternal chromosomes. In telophase 2, a nuclear membrane forms around every set of chromosomes, and cytokinesis takes place in both daughter cells, producing an end product of 4 cells. And that is the process of meiosis.